Thank you all for inviting me. I'm just going to hand some propaganda out that you can be looking at while I talk. So I am the director of the Chesapeake Biological Lab. We are a part of the University System of Maryland, and we've been in Solomons for 90 years. We are in Solomons because our founder, a remarkable gentleman called Reginald Truitt, looked around the communities of the Chesapeake Bay for somewhere to set a lab up to research the declines of oyster. And the people of Solomons took him to their heart and built him the first lab building. And so he established what became the Chesapeake Biological Lab in 1925. I said he was a remarkable gentleman. He was devilishly good looking. He introduced ragtime dancing to the College Park campus. He helped launch the Wright brothers plane when it landed at the College Park Airport for the first time. He took the University of Maryland to their first national championship in lacrosse. He hangs in the Lacrosse Hall of Fame. I have no idea where the Lacrosse Hall of Fame is, but if you ever find it, you'll find a picture of Reginald Truitt. And he was a world-renowned expert on the biology of crabs and oysters. The only thing I have in common with him is that I too am director of the Chesapeake Biological Lab. So I tell all of our staff they should be able to answer 3 questions when talking to people. The elevator speech, the Starbucks speech, whatever you want to call it, is who are we, what do we do, and why is it important? So I'm going to try and do that for you this morning in the next 15 minutes. So who are we? We are part of an organization called the University of Maryland Center for Environmental Science, which is a campus level unit within the System of Maryland. So we are equivalent, but obviously much smaller than College Park, Towson, UMBC, and we are a graduate school. We train graduate students. We provide research training for master's degrees and PhDs. We have no undergraduate students routinely on campus. We have 4 campuses spread throughout the state. We like to say they are strategically located. We have our sister lab on the Eastern Shore in Cambridge called Horn Point Lab. We have a lab on the western part of the state, just off the Frostburg State Campus, which is called the Appalachian Lab. And our newest laboratory is in the Inner Harbor. It's called the Institute for Marine and Environmental Technology. And together, those 4 labs meet our legislative mission. We are the only branch of the university to have a legislative research mission. And our mission is to conduct a predictive ecology for the Chesapeake Bay. And we have no idea what that means. We have no idea who put it in the legislation, and we have no idea what their expectation was. But we interpret that mission to mean that the 4 labs together will conduct research on any aspect of the environment that affects you, the citizens of Maryland, or people who make decisions on your behalf. And so we have research programs that look at the deposition of natural and human released materials from the atmosphere onto the landscape and onto the seascape. We have people who look at climate change in the Arctic. We have people who look at bluefin tuna in the Mediterranean. So we are, as we like to say, globally eminent and locally relevant. So that's who we are. We're a graduate research lab that has this unique applied mission. What do we do? I've given you a little bit of hint of that, but let me give you some depth of what we do at Chesapeake Biological Lab. So we have 4 main areas of research focus. One is fisheries, and I'll come back and talk a little bit more about that because that's my research area. Another one is ecology, and we study all aspects of the ecology of the Chesapeake Bay, from microscopic bacteria and the role they play, to whales out on the coastal shelf and how they interact. We ask who eats who and by how much, what happens to fluxes of material, and what can we do to restore those ecosystem services. We have another group that studies toxicology, and when that research first started, it was really a question of how much of this material will kill. And thankfully, we've gotten long past that question because most of the releases of chemicals are now at sublethal levels. And so the questions we ask are, what are the impacts of this level of a chemical? The third area we call biogeochemistry, which sounds a complex topic, but it really is a case of studying how chemicals move through the environment. And so what chemicals do we study? It's a very, very wide diversity of chemicals, from nitrogen released from septic tanks. So we have scientists who study how nitrogen that leaks out of someone's septic tanks and perhaps goes into the Potomac River, how is it transformed, where does it go, what are the impacts of that material? We also have a researcher that looks at what makes a vintage French wine a vintage French wine.
You ask for volunteers?
Yeah, really.
It is sadly destructive testing. We don't get to have the human factor. It's all a case of instruments and machines. We also study materials like methane and mercury, for example. When the state releases an advisory that says you should eat 1 or 2 meals of striped bass from the Chesapeake Bay and no more based upon exposure to mercury, those data come from us. So we do a range of different topics. And what is remarkable to me is those 4 areas that I discussed were Truett's vision back in 1925. And that's why I think he is an unsung hero of the environmental movement. He had a clarity of vision in 1925 that I think was unsurpassed. And he really deserves a lot more credit and a lot more knowledge of the impact that he had. So I said I would talk a little bit about fisheries because that's my area and it's perhaps what we're known for most. Truett founded the lab because he was concerned back in 1925 for declines in oysters and crabs. Now we think about concerns of oysters and crabs being a very recent affair, but here was this gentleman in 1925 saying oysters have declined and I want to understand how to bring them back. So many of you may know that the oyster harvest in the Chesapeake Bay in the 1800s was substantial. There were something like 16,000 bushels of oysters taken out of the Patuxent River in a single year in 1880. When John Smith came over here, oyster reefs were an intertidal hazard to navigation. And that early fishery was really not a fishery, it was a mining exercise. Right? And there are many towns, including Solomon's, that I like to say are literally built on the oyster fishery. And you can look at historical pictures of Solomon's and see that the area between the sort of Avondale part before the church and the Episcopal church was open wide. There was about 100 yards of open water. There's now open water probably the width of 2 of these tables. The rest is filled with oyster shell. So Solomon's, like many coastal towns, was literally built on the oyster fishing. We've also had concerns about crabs in the press recently. Those concerns go back a long way. And we've had concerns about striped bass as well. And many of you may know striped bass is considered one of the success stories. Right? That The striped bass fishery was closed for a few years in the 1980s, again, largely based upon research conducted at the Chesapeake Biological Lab. It was opened again 5 years later, and striped bass have been resurgent ever since. So I wanted to take a little bit of time to talk in depth about the research that we're doing on oysters, because that's usually the topic that is of broadest interest to people. So oysters play a remarkable role in the Chesapeake Bay. They are what we call ecosystem engineers, in that they change the environment. When you think about oysters growing in a reef, they change every aspect of the environment. We think of them changing the water flow, obviously, because they are a structure in the Chesapeake Bay. But they also change the chemical environment. So you are asked perhaps to pay a tax on your water bill to support nitrogen removal from sewage systems. If you're not on a county sewer and you're on a septic system, if that fails, you're now asked to put in an advanced treatment sewage system which removes nitrogen from your waste from the sewage system. Oyster reefs do that naturally. They are an important structure that removes nitrogen from the water column. And why do we want to remove nitrogen from the water column? Because there are far too many nutrients in the Chesapeake Bay, and that's what causes the phytoplankton to bloom. And when the phytoplankton bloom and die, they settle to the bottom, and all the microbes start eating the dead phytoplankton. They use up all the oxygen, and we get the dead zones that characterize the Chesapeake Bay now. So we have oysters providing structure. They change the chemical environment. They also change the biological environment. If you look at the community that is associated with an oyster reef, it is one of the most diverse communities you will find. So not only is it oysters and other animals like oysters, like mussels, but it is also strange animals called sea squirts that look like grapes. And they're called sea squirts that, if you've not squirted your grandchild with one, you should. It's one of life's great pleasures to pull it out from an oyster reef and say, look at this. They also support a large number of small mobile invertebrates like mud crabs. They are home for blue crabs. Blue crabs love structure. And they also attract a large number of fish because everything else is there for the fish to feed on. So these oyster reefs engineer a different environment. And we have lost vast acreages of oyster reef in the Chesapeake Bay over the last 100 or more years. And so there are many efforts to restore them. And I know the Rotary Club has been involved in trying to restore oyster reefs in St. Mary's River, and I congratulate you for that role. You may well know the state is investing, and the federal government are investing millions of dollars restoring Harris Creek off of the Choptank River. And this is the first time we have ever tried to restore an entire river system. Most of the time we restore small areas, perhaps the size of this room. But we're trying over in Harris Creek to restore 300 acres of oyster reef to provide that ecological function. We don't have the ability to do the engineering to put 300 acres of oyster shell out, but we do have the ability to monitor the impacts of that change. And so those 4 skills that we have, the fisheries, the toxicology, the ecology, and the biogeochemistry, we bring that all together to monitor how that restored reef is performing. So that's the first thing we do with oysters. The second thing we do with oysters is that we try to understand their direct impacts on the water column. So many of you may know the number that an oyster can filter 30 gallons of water in a day. And if you came to the Oyster Festival, you may have seen our display there where we had 2 tanks, one full of algae that looked green, another one that was the same color at 10 o'clock in the morning, but probably by the time you saw it, the water was crystal clear because the oysters had taken everything out. So oysters do a remarkable job. But what most people do is say 1 oyster filters 30 gallons of water. There are 1 million oysters, so there must be 30 million gallons of water being filtered. But what happens is that oysters grow in a clump. So this oyster, if the water is flowing this way, this oyster filters 30 gallons of water containing all the phytoplankton. This second oyster gets to filter some of the water that this one's already filtered. So by the time you get down to this one, all the way downstream, it's seeing clean water going past. So we're trying to understand how the mass of oysters filters the water. And probably the most important thing that we're involved in with oysters at the moment, many of you, if you follow the legislation that came out of Annapolis this winter, you may have know of a piece of legislation that passed House and Senate that mandated that the Department of Natural Resources conduct what's known as a stock assessment, which is an analysis to say how many oysters are there in the Chesapeake Bay. Now that's a number you thought we would have known. We have been working on oysters at CBL for 90 plus years. We have no idea what that number is. Why is that number important? If you want to harvest trees from a forest, you think you would want to know how many trees there are so you know how many trees you can cut down and still have a forest. We don't have that number for oysters. And so over the next year or two, we will be working to provide that number for the state so that the state, on behalf of citizens such as yourself, can say how many oysters can we safely harvest from the Chesapeake Bay without denuding the oyster reefs that are still there. So I'm going to close just by talking about the information that I put out. I became director at CBL in 2011, and one of my first actions was to recommit to engaging with the local community. So those of you who live in the area may have noticed the wonderful signs that cropped up on all of the buildings over the last few years. When I took over, there were no signs on any building anywhere. We could have been dissecting cadavers for all anyone knew. So we decided that we are actually very proud of everything that we do. So we committed to putting signs on every building that tell you a little bit about the history of the building, and then below that, something about the research that goes on in that building. So you can now do a sort of self-guided tour around our campus and find out a lot of what we do. The second thing I committed to do is this Science for Citizens. We have the last one of this season's talks next Tuesday, but we do a series of 5 lectures in the spring on a Tuesday evening at 7 o'clock, and we do a series of 5 lectures in the autumn on a Tuesday evening at 7 o'clock. They are intended, as the name suggests, to talk about science for citizens, and they are offered at a level that is intended for a high school student. That was the audience we were aiming. Unfortunately, we don't attract too many high school students, but we're working on that. You can look at the 5 talks that we have given this year to get some idea of the range of topics that we typically talk about. So we had one, I started off talking about fisheries. You'll see the next one was about mercury. So we've had the fisheries and the biogeochemistry part. The third one was about mountaintop mining in the western part of the state and how that affects streams. The one, what was it, 2 nights ago, was called Manure Happens. It took me a long time to stand up in public and say, manure happens. And it is the consequence of our agricultural system that feeds 7 billion people largely a meat diet, and the environmental impacts of that. And then the last one, which is this coming Tuesday, is not fear the turtle, as College Park would like us to say, but it's fear for the turtle. And it's talking about the impacts of climate change on diamondback terrapins and the impacts that's going to have on their nesting sites, but more importantly, on their sex ratios. So who in the room knew that the sex of a turtle is determined by the temperature at which its eggs are incubated? One or two of you? So we think of sex as something that's just a sign, right? Chromosomes tell you what sex you've got. In many animals, in fish, in many amphibians and reptiles, the sex you have is determined by the temperature at which your nest incubates. So you can only imagine, as our temperatures rise, the potential impacts on the ratio of male and female turtles going forward. So that's the topic of the final talk. They are, I want to make one last comment about these presentations. We are a scientific research institute. Our credibility to the citizens and to the state government and the federal government that we serve is that we are objective, independent researchers. So even though we may have personal views about a range of environmental issues, these seminars are not advocacy. Right? They are a presentation of the best knowledge we have about the topics at hand. The one about manure happens was not a drive to have everyone become vegetarians. It was a simple statement of, here are the challenges that we face in our agricultural system, largely because it leaks nitrogen at every step. Right? What society does with that is a far more complex discussion than the science itself. So I encourage you all to come, if not for this one on Tuesday, for our series next year. And as I said, if there's anything we can do at any point, don't hesitate to ask. Thank you very much.
Thank you.